Hello everyone, I am Dr. Amit. Welcome to my YouTube channel. Please subscribe and press the bell icon. This is our new video in the biochemistry series. Today we'll talk about metabolic sites and difference between hexokinase and glucokinase enzyme. Then from the next video we'll talk about glycolysis, and then in the upcoming videos we'll talk about all the different cycles of the biochemistry. Okay, so it's very important to know about that which cycles is occurring where. Okay, because it helps us to understand the different steps of the cycle while learning the details in the cycles. Okay, so in this video we have let's learn first. There are three metabolic sites, that is mitochondria, cytoplasm and both. Okay, so inside the mitochondria we have fatty acid oxidation, acetyl coenzyme production, TCA cycle, that is Krebs cycle, after then oxidative phosphorylations and ketogenesis. Okay, so inside in the cytoplasm we find glycolysis, then HMP shunt, synthesis of cholesterol that occur in smooth endoplasmic reticulum, then synthesis of protein that occur in rough endoplasmic reticulum, then synthesis of fatty acids, then synthesis of nucleotides. Now some cycles happens in both mitochondria and cytoplasm. Okay, so to remember this, just think of the word hug, HUG. Okay, so this include heme synthesis. So H stand for heme synthesis, U stand for urea cycle, and G stand for gluconeogenesis. Okay, so many MCQs are asked from this part. Okay, so this is very important. So please mug up that which cycles is occurring where. Okay, now we'll talk about the difference between hexokinase and glucokinase enzyme. Now here we'll discuss what's the difference between hexokinase and glucokinase and why it's important to know the difference between these two. Okay, so let's compare hexokinase versus glucokinase. Okay, so before understanding this, let's first understand what is kinase. Okay, so kinase catalyzes the transfer of phosphate group. Okay, so it catalyzes the transfer of phosphate group from a high energy molecule like ATP to the substrate. Okay, so this both in hexokinase and glucokinase enzyme helps in phosphorylation of glucose to yield glucose 6 phosphate. Okay, now the main thing here is this glucokinase. Okay, this glucokinase enzyme work where there is high concentration of glucose, like in liver and beta cells of pancreas, but where there is low concentration of glucose, like except liver and beta cells of pancreas, in other tissues hexokinase works over there. Okay, so wherever there is high concentration of glucose, glucokinase work, and where there is low concentration of glucose, hexokinase work. Okay, but the important point from clinical point of view, okay, if there is glucokinase deficiency, okay, then it can lead to MODY. Okay, so the full form of MODY is maturity onset of diabetes of the young. Okay, and due to deficiency of glucokinase, gestational diabetes also occur in few patients. Okay, now we'll learn the points. Okay, the points difference between hexokinase and glucokinase. Now here is a comparison table. Okay, so we have total 5 differences. Okay, we'll talk one by one. Okay, so first we'll talk about the location. Okay, so as we have already discussed that hexokinase is found in most of the tissues except liver and pancreatic beta cells. Okay, so glucokinase. Okay, so glucokinase is found in liver and beta cells of pancreas where there is high concentration of glucose. Okay, now we'll talk about Km. Okay, so this Km, how to define Km? Okay, so it's a substrate level, or we can say the substrate concentration at which the reaction rate is half of Vmax. Okay, so the Km is lower. Okay, so Km also define about the affinity of the enzyme. So Km is lower for hexokinase. So if Km is lower, the affinity is higher. So for hexokinase, Km is lower and affinity is higher. For glucokinase, the affinity is lower because the Km is higher. It's this Km and affinity is inversely proportional to each other. Now the third point is we'll talk about the Vmax. It's also called as maximum rate of the reaction. Okay, so Vmax is lower. So the Vmax indicate the capacity of the enzyme. So the Vmax is lower for hexokinase and its capacity, so it has decreased capacity. Okay, but the Vmax is higher for glucokinase and it has more capacity. Okay, Now we'll talk about how insulin affect this enzyme. Okay, so this hexokinase is not induced by insulin, but glucokinase induced by insulin. Okay, now we'll talk about feedback inhibition. Okay, so hexokinase is inhibited by glucose 6 phosphate. Okay, and this glucokinase is inhibited by fructose 6 phosphate. So we can remember like this. Okay, before H, G comes. Okay, so G inhibit, so glucose 6 phosphate inhibit hexokinase, and before G, F comes, so fructose 6 phosphate inhibit glucokinase. Okay, so that is it for today. Thank you for watching. So please like, share and subscribe to my channel, and please like, share and subscribe, and in the next video we'll talk about glycolysis in detail. Thank you.